Bom, então vamos dar continuidade no nosso evento, a conexão entre a nutrição e saúde intestinal. E temos o prazer, então, de convidar o Dr. Peter Ferek para iniciar a sua palestra. Então. Yes.
I got the microphone. There it is.
Agora, vocês, quem precisar tem que buscar o fone, tá? E no chat, online, quem estiver online, coloca no globinho o idioma português, se quiser ouvir em português, ok?
Well, thank you very much, and it is a great pleasure to come to Brazil and meet so many families of mine. All right, these are students that have grown up, and Alessandro is one of those, and those online. I'm really thank you for your attention today, and it's a great honor to be here to present. Dr. Fernandez gave an excellent overview of gut health, and she did most of my work. So my presentation will have some similarities of what she has, but I'm going to bring it now to a more practical overview of it, and then show you some new technologies that are developing in my laboratory that is kind of helping with this whole process.
Interpreters, could you ask a little bit
slower? Okay. I need to speak slower. So my presentation will be about feed formulation and maintaining the enteric ecosystem symbiosis. All right, so this is very similar to the theme that we've been talking about today. Okay, I need to stand here maybe. So when we think about the gastrointestinal tract, as Dr. Fernandez pointed out, it's a very complex one. It is loaded with different cells, and we are just now beginning to understand the impact of the microbiome. In fact, many of you have already heard that the enteric microbiome has 10 times more cells than in the entire body of the chicken, and 75 times more genes in the host. That means when we feed our chickens, are we really feeding chickens? No, we are feeding a community, and the chicken just happens to be part of that. And once as nutritionists, we understand that we are the suppliers of the nutrients to an ecology, and the chicken is part of that, that we begin to understand where dysbiosis may occur, not only in the microbiome, but within the chicken itself and the entire environment, including the litter, the house, and us as working with chickens. Dr. Fernandez showed that picture before about this, and this was a picture that was designed by Dr. Ed Moran, who was my master's professor, and I was in the lab when he developed his book about comparative nutrition. And this is the picture of the chicken, and the gut ecosystem and that microflora that is developed there is a consequence of that milieu. It's all the factors that's within the gut. And because the chicken has a very short intestinal tract, its unique physiology in digestive physiology creates zones that are unique. And within those zones are little micro ecosystems where the bacteria, the protozoa, and other cells thrive. And it's influenced by the pH of that environment, the osmotic properties, the anaerobic and aerobic nature of that environment, the gut motility, as Dr. Fernandez pointed out, and as well as the nutrient composition of that digesta. All of these factors influence that ecosystem. So this is what a good healthy gut should look like in a chicken. There should be a transition from food, this is the crop, and all the way through. And as it's progressing through, nutrients are digested, and all of the undigested materials, predominantly the cellulosic type ones that produce volatile fatty acids, are developed in the ceca. And what you want to see is the ceca goes through, it's a fermentation vat where it loads up, ferments, and then completes and expels and goes through that same cycle. But when you have digestive problems, you can have issues. Now, the functions of the gut microbiota are many. First, it has a metabolic function, and that is it will take dietary components, and the balance of those dietary components depends on what microbiota then will thrive. So it could be proteins, carbohydrates, etc. They produce metabolites, organic acids. They can produce vitamins if you've got the right microflora. They'll deal with some byproducts of metabolism like hydrogen, CO2, and methane. And also, these co-products can be metabolized by the host. So there are certain things like, for instance, methane. The methane actually is produced by methanogenic bacteria, but just methane by itself can actually change the gut microflora and also the motility of the gut. So methane has an adverse effect, slows gut motility in a chicken, or changes it. And the next, it could have, let's see if I can advance here. Sorry, my advancer doesn't work here. Here we go. It has a protective function as well, where the mucosa or the organisms can produce antimicrobial factors that control the microbiome, but also displace certain pathogens that are competitive. And it can have an immunological impact on immune system development. All of these things was described by Dr. Fernandez very eloquently. But the next thing we're starting to realize is that it can also have a neurological effect. In fact, many of these microflora produces dopamine, serotonin. These are neurotransmitters, and they literally affect the endoneurological systems through the parasympathetic nervous system in the gut, and that's conveyed to the brain and affects behavior. So when you've got chickens that have an enteric distress problems, they will behave differently. They'll be anxious. They'll have behavioral issues where it disrupts them, and so it has an animal welfare component. So these are the new things that, especially when we're concerned about animal welfare, that the gut microbiome now becomes a part that maybe helps with the welfare of the animal. So when you have enteric distress and inflammation, this is the good gut in a normal condition. You have the microbial community that develops, and you have tight junctions that prevent these organisms from translocating, as Dr. Fernandez pointed out. But when you have stress, and that stress could be environmental, it could be mycotoxins, it could be bacterial dysbiosis, viral infections, a number of things, off feed, any type of stress, and I even put down here psychological stress. I mean, we had a situation in a trial where we had a fan that was squeaking really loud and in one room, and then we had another experiment where in another one, and the ones they were fed exactly the same, but the one in the very loud squeaking fan room, they had diarrhea, they had all kinds of issues. And so it could be something related just to the environment. And so when you have these sources of stress, you can run into situations where these tight junctions open up, and then the translocation of bacteria, it initiates an inflammation, and you end up with inflammation that compromises digestion. Dr. Fernandez really went through and described that eloquently. And so when you have a sick microbiome, all of this stuff ends up in the hindgut. And this is a picture of the ceca. And you end up with very aggressive fermentation, producing gas, putrefying proteins, etc. And all of that now causes inflammation. And when that happens, there are neurological signals that are sent to the foregut to say, okay, it's creating malabsorption. And the reason is that these organisms in the hindgut want food. They want starches, they want protein, they want fat, and those are the pathogenic ones. And they send signals to the front of the gut, don't absorb these things so we get more food. And that's really, it's who gets fed is the big issue. So I'm going to describe to you just the ecological perspective, and I'm stepping back as a poultry nutritionist, and I went and started reading all the ecology type studies. And if you go into the classic ecologies, whether it's plants, a forest, or something else, there are three factors that influence that ecological development. The first is to establish an ecological environment first and cultivate that physical ecosystem. So, for example, I'm from Canada, way up north. There's not too many plants. It's cold, it's tundra, and there's the ecosystem is very fragile. You spill oil or do anything in that environment, and for generations it destroys that ecosystem. You have the Amazon, very lush, very robust ecosystem. Okay, you drop some oil or some kind of contaminant in the forest, and it's all eaten up by something, and it rebounds quickly. This the structure of the ecology that flourishes and makes whether an ecosystem is very diverse and robust versus one that is very sensitive. So that's an important part. And then finally, the other part is seeding symbiotic bacteria. So you want to seed with the symbiotes that maintains that ecosystem and exclude any invasive species, all right? So one of the things we have, like in North Carolina, somebody brought in kudzu. It's a plant. They thought it would be really nice because in China, they were feeding it to cattle and they thought, well, let's put it. But there's no natural predator for kudzu in North Carolina. So if you drive down, you see this mountain leafy material just overtaking everything, and it's disrupting the environment. So that's an invasive species. So when we're thinking about the animal, we also are battling with invasive species versus ones that are symbiotic. The next factor is the nutrient balance. And the idea here is to feed the symbiotes and starve the pathobiotes, right? The pathogens. And Dr. Fernandez talked about that. And then finally, how do we maintain that symbiotic stability in that ecosystem? And you do that by controlling the diversity of that and the microbial load, how much there. Because if you end up with too many of the same kind of organisms, they turn into an angry mob, and they start fighting for resources, and they become pathogenic and send signals. And then finally, let's weed out waste. So we're going to cover each of these factors in some way. I grew up on a farm. My family farm, we grow corn, soybeans, and as I was driving here, I looked at all these beautiful fields of soybean and corn, and we also had pigs and we had some poultry. And my father got his degree, what is now Wageningen Agricultural University, but it was there when it was a college. And he said these are the 4 factors to get a good crop of anything, whether it's plants or chickens or whatever. You have to cultivate at the right time, prepare the land, put in good seed, feed at agronomic rates, and then weed out the bad stuff. And then you pray for rain, for God to bless you with the rain. So we're going to go through each of these same kinds of concepts, but this time, how does this work, these principles within a chicken? And the first example I'll give you is related to perinatal development. This is setting up the ecosystem so that its environment is now capable to accept the right conditions for that animal. And some really good research was done years ago now, but here's a list of them. That vertical transmission is a conserved mechanism to inherit the commensal or microflora in several poultry species. And Procedo, another good Brazilian, identified salmonella in the ceca contents in 18-week-old chicks, day-old embryos. And she used these in situ hybridization to demonstrate that. Before that, many people thought that the embryo was quite sterile, but it's not. And one of the ideas here is that if you can develop a very good microbial ecosystem in the hen, that is going to transfer to the chick, and that is incredibly important. So when we think about our commercial poultry production, we've actually removed the hen from the chick in many ways. We feed restrict them to control their body weights and all these other things, and then we try to sanitize everything, and so the likelihood of passing on a good commensal organisms is limited. So here is an opportunity for probiotic treatments of your broilers and actually putting technology in the broiler breeder so that it will pass on some good commensal pioneer colonizers for that chick. And just remember where that egg comes from, all right? It's being laid, but it's going through the cloaca, and that cloaca is the junction of the cecal droppings, the feces, the urine, and it's laying that egg, and as it's doing so, any bacteria is going to be, whether it's good or bad, is going to be locked into that cell structure. And the cuticle is wet, and as that egg is dropped, these organisms become locked into the shell, and then once the cuticle dries, it's locked in there. So these are your primary colonizers for the chick. And there's also some microflora that will migrate up into the uterus and things like that in the chicken that can also impact that, as well as into the egg itself, just as Prosoto demonstrated. So we looked into this further, and actually before Prosoto, I had a graduate student, Bojorquez. He's a student from Ecuador. He's now a professor at Duke University, and he's doing mostly his work with neurobiology. He was the founder of the gut-brain axis movement that's going on now. But during his research, we were doing a survey of broilers, turkeys in this case, and you know, a turkey hatches at 28 days of incubation. And this is the ceca. And we noticed always in a lot of these electron scanning micrograph, these organisms. And as we zoomed in, we can see that it's already a variety of different organisms that exist. This is by 19 days of incubation. That community becomes a little bit more complex. And by the time they hatch, we can also find that when we used some more profiling of data, this is the original of looking at microbiome profiling, we identified a lot of organisms that are non-culturable, but a whole list of organisms of lactobacilli, bacilli, and pseudomonas. And when we went to go sample the breeder hens from these chicks, these were also the dominant type microflora. So we know that what is actually in the bird before it hatches already has these primary colonizers. Dr. Bojorquez also worked with me on a concept called in novo feeding. And this is the idea where we're supplementing nutrients into the amnion of 17-day-old broilers. This is right about the time of transferring from the incubator to the hatchers. And for turkeys, we can do it at about 24, 25 days of incubation. And this is at the time when the amnion surrounds the embryo, that the embryo will swallow, that's their first meal, will swallow that and begin to consume it. And that initiates the metabolism ready for hatching. So and basically the idea is like we supplement that amnion like it's his first meal, much like we put a premix in corn and soy diets to actually help balance the nutrients. And that's basically what this does. And so these formulas contain a variety of different B vitamins and certain kinds of compounds that we found to be most important for early development. And we found improvements in glycogen reserves, better chick quality, advanced gut health development, skeletal development. We see increased body weights and gain as well. Muscle yield because it enhances the satellite cell development and breast muscle. And even when these breast muscles are being challenged because of woody breast and spaghetti breast, it's those satellite cells that replenish and heal it. And if you don't have enough satellite cells, this is one of the reasons why we see the development of these conditions. And it also enhances immune function. But I want to get to really the heart of this technology, of how it influences the gut microflora. So here is, these are intestines of turkeys, again, from Bojorquez. He was really good at electron microscopy. This is a control bird. This is the villi of the jejunum. And you can see the tips of the villi. And in the control system, they're not very well developed, where the microvilli are very short. And so that's why you can see the outlines of the enterocytes. And the gaps between these are not completely closed, so they're actually open for translocation and any kind of microflora that may happen there. Whereas the novo-fed birds are far more well-developed, microvilli long. They're producing mucin that provides a protective barrier, and it actually helps with advancing the digestive capacity of these chicks by about 3 days. So if you can imagine, a novo-fed bird at hatch has the same enteric development characteristics of a 3-day-old chick that did not get that. So it has a huge impact on the ecology. But if we look at the ileum, this is at the very end of the intestinal tract, you can see a huge difference by in novo feeding, where here you have the control. They're somewhat dehydrated, not well developed. In novo fed are very well developed, and you start seeing the colonization of these long filamentous short organisms. Dr. Fernandez showed you some pictures of those organisms. They colonize along the way, and they're the ones that that produce short chain fatty acids, very rich in butyrate, and butyrate is necessary for early enteric development, but also helps with the colonization of other good bacteria like lactobacilli, etc. And they also stimulate the immune system and mucin secretion that provides a protective barrier. So we didn't put any probiotics or prebiotics in the formula. This is just adding something that mostly it was a little bit of oligosaccharides, and we had some B vitamins, predominantly folic acid. And if anybody wants to know all the details, I can provide you the formula for that. But we do see a significant impact. Dr. Fernandez also showed you this picture, but where she was talking mostly about the impact of early nutrition on enteric development, what I want to emphasize is there are 2 time periods where you can program the gut ecosystem of a chick, predominantly the primary or the pioneer colonizers. The first window is at 12 days of incubation. And in this case, you can do that by injecting right into the air cell prebiotics like galactose oligosaccharides. These are the oligosaccharides that are the product of, let's say, a xylanase. So you can have acyl-oligosaccharides, a mannan-oligosaccharides, a galacto-oligosaccharides. So these are the short chain oligosaccharides that are prebiotic, and that stimulates the microflora. But you can also put a probiotic at that time. The next window is during that in ovo feeding period, as we had demonstrated before, and you can also add in a prebiotic in that case. What those 2 conditions happen is when that chick hatches, it now already has not only the primary colonizers that is coming from the hen, but also prepares the gut to accept and establish a more rapid development of the ecosystem. So during that first 14 days is critical. And if we can get a jump on establishing a good, stable ecosystem, even by the time they hatch by this technology, we will really advance the capability of making that bird more resistant to any kind of enteric pathogen. But of course, as Dr. Fernandez pointed out, there are lots of other things that can impact the development of the ecosystem. The next thing about cultivating and seeding is the idea of using a probiotic into the treatment. And this is a technology which is being developed pretty rapidly, and many companies are using some aspect of probiotic treatment in somewhere. Many of these are culturable organisms, all right? So many of the things that I showed you with the in novo applications, those are non-culturable type things and can't actually create a probiotic from that. But here, Lactobacillus, Bifidobacteria bacillus, Enterococcus. Here's one that I worked on a while ago. It's called Clostridium butyricum. It produces a lot of butyric acid, and it helps with development as well. So there are a few species considered for use. Obviously, you have to be concerned about the stability, feed processing, stability within the gut, all those other factors that's involved. And I classify yeast as part of that microbial probiotic type concept and their constituents, because there are also fermentation byproducts that can help with that ecosystem. So one probiotic that has gained a lot of popularity and flexibility is the bacillus type strains. And I wanted to go through the various mechanisms of how a bacillus strain happens and can help. You can select these bacillus strains for a variety of different attributes. And this is one of the reasons why this technology is really helpful by combining it with other things like an enzyme, because you can now select an organism that thrives in the presence of an enzyme and help contribute to the development of that gut. So the first step is that these organisms can colonize, and as they're passing through, they produce some enzymes. And some of these enzymes are beneficial digestive enzymes. And these nutrients then can be readily absorbed because they are colonized right along. They can also produce other compounds that actually competes for nutrients for these pathogens. So these pathogens want to use protein, fat, etc. But with the bacillus, they're their activity is a little bit more aggressive, and so they'll be able to take those nutrients, help digest it, and so it can be absorbed by the host. And the other thing is that they can produce metabolites like volatile fatty acids and some antimicrobial peptides that will inhibit some of the pathogens that are competitive. They can also compete for space in adhesion sites, particularly within the brush border and the mucosal lining as well. And they also produce other compounds, quorum sensing or compounds and things like that, that will modulate their other friends. Some will be make them some bacteria happy, others indifferent, and some will actually discourage their development. So depending on what kind of bacillus strain you develop and select, they have a variety of opportunities. And so with each strain, you can enhance or decrease any of those properties to be functional. And this is some research that was shown by Smirnov. And this is, I was working with Dr. Zahava Uni, and she was a student of Zahava Uni, so I was participant of this research in her PhD. And we looked at the goblet cells and the effects of probiotics and antibiotics on the villi structure of the gut. And this is in the ileum or the jejunum. And you can see with the antibiotic, it reduces the microbial load. And because of that, it doesn't need much barrier function activity to help. So this is why we see better feed conversion, is because you're not maintaining such big tissues and you're also not producing a lot of mucin cells. This is the control but if you add a probiotic, the opposite happens. It stimulates a lot of mucin, and actually the villi are increased. Obviously, what the probiotic is doing is enhancing the barrier function, but it comes at a cost. All right, so you may not see feed conversion benefits. In fact, sometimes adding a probiotic, you may see the opposite early on. You may see in the pre-starter period that feed conversion actually goes down. And that's because it's got to support not only that mucosal barrier, but it's also got to secrete a lot more mucin. And the amino acid profile of mucin is very different than the amino acid profile of muscle. And when you put in a probiotic, it has a mandatory requirement, obligatory requirement to satisfy the mucin, not the muscle. And so if you don't have enough proline, threonine, and some of those amino acids that are rich in mucin, it could affect muscle development. All right, so this is one of those things when you add a probiotic or do something that really enhances that brush border, you've got to make sure that your formulation is set to be able to supply that need. Otherwise, you're going to have compromised in performance. The third example that Dr. Fernandez pointed out is a function of gut motility. And this is a concept that we worked on for many years, I together with Dr. Brake, on feed particle size. And as you can see, you've seen this picture before, but the particle size is very important for that gizzard function. Here you can see a gizzard that is fed coarse grind whole grains, for example. It has a very strong muscular characteristic, and the glandular stomach, the proventriculus, is relatively compact. But if you fine grind the same formula, you end up with a very small gizzard muscle because it doesn't have to work much, but it creates a situation where the proventriculus now has to grow quickly because they only have one shot at adding digestive juices. And so you end up with hyper production of gastric function, and sometimes you'll have lesions there, and then they'll cause some problems. So when you have a coarse particle, Every time the gizzard contracts, material goes back into the proventriculus to get another shot of peptic digestion. And it's very critical. Pepsin is an endopeptidase, all right? So it clips a protein in the middle. And so now it doubles the size of the the ends, which is important for the trypsin and chymotrypsin, which are endo. They chew amino acids from the end. So it doubles the efficiency of peptic digestion in the small intestine. Every time that gizzard contracts, fine material also goes into the duodenum, where it is mixed with the bile as well as pancreatic lipases and et cetera, and proteases, and it goes back and forth. And every once in a while, depending on how much fat is in the diet, there'll be reverse peristalsis all the way back into the gizzard. And that's why the gizzard is yellow color, because of the bile. And it's very important for fat digestion. So two things are really important for good gizzard function: protein digestion and fat emulsification and digestion. And those are the two things, if you don't absorb it, these compounds in the front of the gut, they end up in the hindgut. It feeds the pathogens. You also have reverse peristalsis that would enhance digestion. And the way this works is as nutrients are being absorbed, it will keep going back and forth until that bolus now is not producing the nutrients anymore, and then it passes on through. So if you have something very viscous and it can't absorb the nutrients that's trapped in there, the chicken says, well, why do I have to work on this one so hard? It's costing me too much energy. I'll just pass it on through. So this is why a xylanase helps if you can break down that glob. And then finally, you have this low amplitude reverse peristalsis that's coming from the ureter excretions, non-protein nitrogen. If you notice that the ceca are pointing backwards, that means it's got to go this way and then up into the ceca. And this is where all the undigested material, and particularly the small particles, and if you can get the cellulose and some of those oligosaccharides into that ceca, together with the non-protein nitrogen, this functions much like a cow's rumen. And it produces volatile fatty acids. And those volatile fatty acids not only inhibit pathogens in the ceca, but also provides a considerable amount of energy now that we know. And so it has a particularly important concept there. So gut motility is critical. So the idea is, how do we control that? And so when I was in the Netherlands, I did a sabbatical there. Wow, this was already in the mid-90s. This is where I was looking at, they were adding whole wheat to the feed. And that was just because if they used domestic wheat, they got their subsidy. If they had to send it, exported, then they didn't get the subsidy. So what was happening is the farmers were making a deal that if you take my poultry litter, I'll give you some wheat. And then they started blending, and the feed companies got all upset because it was diluting their diet. And so my job was to go talk to all the poultry industry and say, is this so worthwhile? Am I reducing performance? And then what I found out was actually they were getting better performance, better litter quality. And that was where we all started with this. So I was working with Renee Kwakkel there and others. And that's where that particle size concept developed. This is recently where we said, well, can we put in textures? This is a pelleted feed where we added coarse ground corn to get the same concept. So I had a student, Frank Jean. He's now the nutritionist for the world's largest poultry company in China now. But he did a number of studies, and this is one that is a good example. These are the particle size distribution of a complete feed. One has only the fine grind corn, which is like the conventional way of grinding The other one is that we added 50% of the corn as roller mill. And we found out that if you had a particle size of about 8% of the diet bigger than 1,200 microns, that's enough to provide enough stimulus in the gizzard to have an effect. But if you went more than that, we discovered it slowed down feed intake and it starts affecting growth performance. All right, so there is this, you don't want to overdo with the course. So we took these two diets and fed them to broilers all the way through market. And here's an example of that results. We've seen better body weight at 42 days, feed conversion, an amazing 10-point feed conversion difference. We see the gizzard much larger relative to the proventriculus. The pH of the gizzard digesta was a little bit lower, so we got better protein digestion. Retention time increased because you had more reverse peristalsis, so more time to absorb the nutrients. And as a consequence, we had, pushed the wrong button. We had a better nitrogen retention, better energy digestibility, and amazingly, a lot less litter moisture that came through. After this research, we've done a series, there was about 3 other PhD students that worked on that. Our entire industry quickly changed to doing things, and now we have fairly sophisticated feed companies where the hammer mill tip speeds monitored together with NIR to determine particle size, so that depending on what corn you have, it will now give you exactly the same target of what particle size should be. And it's really had a major effect on performance. But what we found was most interesting was the impact on the gut microflora. Realize that these two diets are identical in their formulation. The only difference was how the particle size was. This is the control. You can see quite a few, about 36% of that gut microflora is within the Enterobacteriotes class, which is considered to be where the E. coli, the pathogens that reside. And you have pretty large amounts of some of the other type of organisms, whereas in the diet that had coarse grind, we actually have a much more diverse gut ecosystem, so more resistant to instability, but we also have less of these bacteriotes and actually more of the more beneficial. You'll see here Clostridioles, 16%, looks like it's a little bit more, but not all Clostridia is bad. There's quite a few that are good, especially the ones that produce volatile fatty acids, like the Clostridium butyricum, for instance. The next step is feeding at the right agronomic rates and weeding out pathogens. And this is where enzymes play a very critical role to augment the digestion of the gut. We have phytases, which are anti-nutritional functions that will cause inflammation, in some cases irritate, and you'll see more mucin secretion. Proteases increase the protein digestibility, can deactivate the protease inhibitors, and even the question about bacterial toxins might be involved in that as well. And then you have the carbohydrases, or the NSPases, that will increase the nutrient digestibility, reduce digestive viscosity, and produce oligosaccharides that are beneficial to the gut. Realize that each enzyme has its own property, and remember, in the gut, there are different places within the gut of different pHs. And enzyme functionality really, and the speed of that enzyme in the reaction depends on the function of pH. So there are several factors that influence an enzyme's functionality. First is the solubility and of the dietary substrates, or the source of the fermentation properties as well. The digestious solution pH, Optimum is anywhere from 2 to 4.5 because that starts in the upper digestive tract. The biocatalytic reaction, this is back to the enzymology. What is the Km, the speed, and the velocity of that? How fast? And this is how it interacts with the substrates and turns around. So if you have an enzyme that has a low Km or a low Vmax, it's going to take a lot more enzyme to actually have an effect. You'd also want a pepsin-resistant type enzyme and one that's heat stable. And it is stable within different moisture in the feed processing conditions, and how that mineral concentration affects the solubility, because all enzymes are proteins, and when you add a cation, it can change the structure of that protein and then consequently the effect. And what other synergistic effects with feed additives? So for instance, there's a synergy that we're starting to know between enzymes and the probiotics and things like that. So in terms of the pH, here is different enzymes that were tested in terms of its pH amount. And you can see that on the left-hand corner is their activities relative to 100%. And so you can see here that some enzymes are functioning very well at low pH, but really drop off later. So this one might be really good in the proventriculus and the gizzard, but not very good in the rest of the gut. All right, so very limited. You have others, for instance, that function only at the higher pH, but very limited. But if you had an enzyme that has a broader combination, or better yet, you take one that has a blend of something that works there and one that works here, you can now create an enzyme blend that has effect at different locations throughout the gut. So how you design an enzyme and its activity, the speed of that enzyme, those are all critical factors in their efficacy. The other component is what's the makeup of the substrate that these enzymes are going. And here are some NSP contents of different compounds. You know, rice, wow, one of the reasons why rice is so good is that it has very little non-starch polysaccharides, whereas something like barley has quite a few insoluble and soluble xylans as well. So these soluble xylans have high anti-nutritional properties. They increase digestive viscosity, and the insoluble ones are highly branched, and they're found in the cell wall structure and prevents the access of nutrients. So a good enzyme has the capability of dealing with both of these insoluble and soluble. So pick an enzyme specifically that can go after these highly branched type structures so that it now opens up and creates it into something that's more soluble that can then be broken down. So the basic concept of most of these NSPs, if it's a soluble compound NSP, it'll form a gel that locks in these nutrients, but even insoluble ones can lock nutrients into a cell structure. And the treatment of that enzyme will liberate these nutrients so that they can be absorbed. And the oligosaccharides that are produced then go to the hindgut and feeds the good bacteria that produce volatile fatty acids, inhibit pathogens, and also provide energy. That's the best of all these worlds. And I want to demonstrate the impact of what we are feeding. So for example, here's the influence of improved ileal digestibility on the substrate available for the cecal microflora. This was research done by Anderson and Stedfeld. And here, if you had a starch, it's already pretty good, 88% digestibility, and you add a xylanase, and so you have better access, that improves that digestibility for the animal to 97%. Pretty good. So it benefited the host a mere 10% of that starch. That's not a whole lot. But if you looked at what's left over, it affects the hindgut microflora it's a detriment by 75%. So the point of these enzymes is not so much to liberate more nutrients, it's what it does to the hindgut fermentation. It has a much bigger impact. Same thing for organic matter, and if you look at proteins, you also see similar trends. So the concept of using an enzyme, though it has a nutrient uplift value, it can have a significant impact on gut health. Here is a study by Anel Santos, another good Brazilian student of mine, and did some work with xylanase. And in his case, he looked at the ileum mucosa. This is the same diet. The only difference was the addition of a xylanase or not. And you can see that without it, you have blunted structure. In fact, it's not that we're losing the structure, is just because these cells are sensing irritation and the microvilli filaments contract. It's like a muscle, and they actually pull down the villi so they look square end and then they also have much more goblet cells to try to protect it. Whereas with the enzyme, you have less irritation, much longer villi, greater capability of absorbing nutrients. And the impact of an enzyme can be quite significant on the gut microbiome. This was a study where we looked at turkey toms. This is 16 weeks of age, okay, so they're quite adult already. Turkeys fed a wheat-based diet, a corn-based diet, and we looked at the dendrograms of the microflora similarities. And you can see that, okay, they're different, but and the diversity indicated by the length of this arrow is low in a corn and much more diverse in wheat, which has more non-starch polysaccharides. But when you add an enzyme associated, and these are enzymes that are appropriate for those substrates, you can see a bigger change in the gut microbiome than you do with just the combination between these two different ingredients. And you also see increases in the diversity, which indicates, as long as you don't have a huge microbial load, the diversity actually inhibits the colonization and makes a more stable gut ecosystem. So enzymes has a great impact on that microbial ecosystem. Dr. Fernandez also showed you this picture that I produced about, here in this case, in 2008. And that's the concept of digestion of fat, protein, and starch. When that's not happening very well in the foregut, those nutrients end up in the hindgut, and it starts feeding the pathogens because they need those same nutrients. And so any source of malabsorption will actually feed more hindgut bacteria to produce these competitive organisms. And I would say also, once we started looking at when you have more of these nutrients getting in the hindgut, it causes inflammation, more mucin. That cost comes at a metabolic cost for that. But what we're finding out also is vitamins and minerals can do the very same thing. Realize that pathogens need these vitamins. Biotin, for instance, is one that's really important for Clostridia, needs it to produce their toxins that actually make them pathogenic. The virulence factors in many of these organisms are dependent upon certain kinds of nutrients, like iron. For instance, Salmonella loves iron. And so if we have too much of that stuff washing into the hindgut, you are feeding the pathogens as well. Whereas symbiotic bacteria actually produce vitamins, right? Just like in a ruminant cow. So knowing this concept, this is my latest patent that we developed a few years ago, is can I create a premix that I can slow release or control release the nutrients. So in this case, we have vitamins and trace minerals encapsulated in a matrix of saturated fatty acids, as well as a few other kinds of compounds we can put in that matrix, so that I can now release those nutrients wherever I want in the gut. So if I want something to be absorbed closer to the front, I put in a little bit more unsaturated fatty acid, make it softer. If I want something that will deliver to the ceca, it has to be a little bit more resistant. So that matrix formulation can be developed that way. And also, the other thing that this technology does is it locks in these vitamins and trace minerals, where a trace mineral will degrade the vitamins if it's left. But if you lock out the air, the oxygen, and the light, and all those kinds of things in a product like this, you end up with stability that can last for a long time. We just recently did a study where we had a vitamin mineral premix made from this sit on the shelf for 5 years and fed it to some broiler chickens, and we still got the very same performance. And we analyzed the activity of those nutrients the same as the day we made it, which is quite phenomenal with this kind of technology. No other premix can claim that. And so the other thing is, because it's in a beadlet form, no dust, no losses, better handling, etc. So maybe I'm biased, but I think it's transformative type technology. And this isn't just an example of what that premix looks like. This is a conventional premix and a microscopic view of it. And this is the microencapsulated. There's the vitamin, there's the trace mineral, but you could put both together within the same beadlets. But this is what the beadlet looks like under a microscope. And we did some studies where we thought, well, we're going to now feed 100% level of the vitamins, and this is what we use the Avigen enzyme supplementation level, and then we fed 30%. And the reason we came to that is we estimated, based on some biological functions, only about 30% of the vitamins are actually absorbed. The rest are wasted, and it has to be metabolized. So we microencapsulated it and looked at it, and then we did it on the free. And if we looked at the performance at the 30% level encapsulated, we got better growth performance. But look at the feed conversion, whether it was 100% level or even at the 30%. That's 70% reduction in vitamins in there. We actually got better feed conversion, and I would attribute that to what happens to the gut microbiome. So, and that's exactly what we found out when we looked at the microbiome of a standard premix and one that was encapsulated. We see that there are more firmicutes to Bacteroides ratio, and that's been shown many times to improve and be associated with better feed conversion. And if we drill, that's in the phyla distribution. And if we move into the more of the genera, we see that we have an increase in the symbiotic commensal microbiota. So we end up with better healthy gut, and this is in actually this was in the ileum. So that technology is something that's emerging. It's not commercially available yet, but it was just licensed by Jeffo. So someday you'll probably see that technology become available to the industry. The next one is looking at for supplemental enzymes. We've already covered that, and some antibiotic essential oils and organic acids. So there are a variety of different kinds of herbs and phytochemicals that's becoming available. Many of you, I worked with Oligobasics, which comes from Brazil. I know many of you use that. That falls within this class, and in that you can now inhibit certain kinds of organisms from developing, reduces the microbial load, and helps assist with these compounds. And the acidifiers and organic acid are helpful. So acidification helps with gastric proteolysis, which is so important, mineral digestibility. It has some antimicrobial effects particularly on the gram negative bacteria, and it supplies energy to the mucosa and helps with enteric development. And this is in a way to supplement those organisms that produce the volatile fatty acids to actually help with that situation. Dr. Fernandez also looked at, gave you some information about the development of the microbial ecosystem. And she said that in the first 2 weeks, that the ecosystem of the gut microflora is slowly developing, but by about after 2 weeks, about 3 weeks, everything begins to stabilize, and this is the volatile fatty acid that's produced in there. So you know when you reached a stable gut microflora, you end up with the metabolites of that microflora become stabilized. And when you have an unstable one, you have these byproducts of fermentation fluctuating a lot and causing distress. But if you look here, right in the beginning, you'll see that butyrate is incredibly low. And when it increases, it corresponds with the development of acetate, which is a major ketogenic enzyme, and propionate, which is important also for the gluconeogenic type. Right? So that's a precursor for things, and it also helps with the development of the intestinal tract that actually enhances that. So the key here is that we want to promote the development of not only organisms that produce butyrate, but maybe even supplement with butyrate is a possibility. Remember in my first slide where we did de novo feeding, you've seen those long filamentous organisms? They produce butyrate, but they're very sensitive to antibiotics. If you treat with antibiotic in the first few days, you wipe those cells out and they will not produce butyrate. So here's just some studies where we used a protected butyric acid, microencapsulated in that case, and we can see not only improvements of the villi structure, less inflammation, but we also see that in the villi tips, you have less problems with the microvilli clumping, which could compromise nutrient absorption. Here's another product that combines essential oils and organic acids. Much of that has got more butyric acid, and we can see improvements in the villi structure as well when we're looking at these additives. And we can see that in this case, here Profeed is a fructooligosaccharides, right? But if we were to put those things in, we see a decrease in certain pathogens. In particular, this is the Clostridial organisms. And this product, Galenet Press, is a protected product with essential oils as well as organic acids. And you can see it has a similar effect. And if we look further into these two compounds, we can also see a significant shift in the virulence factors of these organisms. In this case, it's E. coli. And you can see that with the addition of the protected organic acid and the essential oils, we actually reduce these virulent factors completely or quite significantly. And finally is the idea of mannan oligosaccharides. These are the cell wall structures from yeast. And there are a variety of products out in the market, but most of the new ones now go through a process where they eliminate the linear portions and they extract out that branched function. And so that these branched functions have more mannose units that's exposed, and those more mannose units now have a higher capacity for binding type 1 Formulation bacteria, but also affecting the gut receptors as well that can influence and reduce inflammation as well. And so here's just an example. In this case, this was courtesy of Alltech, but they can bind the type 1 Formulation and cause some sort of agglutination. Each different type of company will claim different things, but the main thing is to test the efficacy could be related more to the agglutination because that gives you an idea of its functionality as function. And this is some research that came out of my lab as well. We were actually the ones that developed the mannose-rich fraction in a study with Altec. It became Actogen. I didn't get a penny out of all of that research, but the impact was quite significant. And in this case, we fed, this is a corn soy diet with monensin, and these are the 14-day-old turkey poults in the ileum, and this was with your mannose-rich fraction. And notice we've got more of these filamentous organisms that produce butyric acid. You don't see any of that in the control. So it changes the gut microflora as well and helps protect. So its function helps with the protective function. It improves mucin secretion. We get a better stable microflora ecology, reduces inflammation, and consequently you get better performance. The last little item, and this is a new venture that many companies are going in, is dealing with quorum sensing. When organisms grow, and this is in case a gram-negative single species, quorum sensing, when they're in low density, there's not much of them. They're just benign. They don't do anything. They live there. They're part of the ecosystem. But once they become getting some nutrients where they develop, they end up with higher density. And some of the stimulus, for instance, salmonella, that could cause this increased amount is biogenic amines and certain other, and methane and those things that actually stimulate the development of salmonella. And once they get really packed in there, they form a biofilm, and then that community becomes more pathogenic because now the issue is they need more nutrients. And when they become pathogenic, start punching holes in the gut lining, the gut starts leaking, they get lots of food, and you've got pathology. So one of the ways, the signals that tells these organisms my nutrient availability is becoming threatened, let's turn on our virulence factors, are quorum sensing molecules. And these quorum sensing molecules have a structure that look very similar to mycotoxins. And so taking clay and certain things that have the capability of binding, you can now select compounds that will have unique binding properties for these quorum sensing molecules and prevent the formation of this change in the nature of these organisms. And so there are products now being developed. Here's one from Amlan International, but they collected a number of prototypes here, but they are ability to bind some of these lactone type compounds, and because they can absorb, they can actually enhance prevent this quorum sensing effect happening. So it's a way of preventing the overgrowth of certain kinds of pathogens. So I conclude with this presentation is just looking at what are some of the things when we're thinking about all of these different compounds. Think as an ecologist, and what additive functions in each of these, because if they're competitive, you end up with one outdoing the other. You want something that works synergistically, and that is establishing the ecosystem environment, promote really good enteric development, and gut motility is important. Enzymes helps with the nutrient balance, and also maintaining that symbiotic nature by weeding out organisms such as antibiotics can help, essential oils, mannose oligosaccharides, dealing with quorum sensing disruption, and also bacterial toxin absorbance are now another feature that becomes interesting. I am the director of the Animal Health and Nutrition Consortium. I have a new project manager now who's an immunologist, Dr. Matt Kosey. And with this organization, companies like BRI and others can join as a membership, and that funds research that benefits everyone. We also have industry companies that are members for free. So Tyson's and everybody's joins it for free, and they sit at the table and talk about the issues, and the other companies now who have something to sell fund the research that supports graduate students. And these are research projects that floats everybody's boat, all right? These are pre-competitive projects. And so if anybody in this room, whether you're supplying or your company, if you're a production company, you can join this for free. Just let me know. And if you're a company that has something to sell, you can now also join with the membership, and that supports their research. We have so far funded $1 million worth of research, and a lot of the stuff I talked about was actually funded in part by this organization. So your participation really helps us with our research as well. Thank you very much. I'll be open to any questions.
Ok, I will ask. Nós temos duas perguntas pelo chat que estão em inglês. E se vocês quiserem fazer perguntas também, está disponível. Eu vou ler essa primeira pergunta, professor. Pode a tecnologia de microencapsulação ser aplicada para probiótico ou enzimas exógenas? Quais seriam os desafios deste tipo de aplicação?
Yeah, you could, and we're thinking about that. Most of the original research is interference in my brain here. Most of the research that we've done was on nutrients, right? Because we want to now affect the release of nutrients. But the other interesting thing is enzymes, and we're thinking about amino acids too, because tyrosine and lysine produces cadaverine and tyramine, and these are both biogenic amines that impact pathogens selectively in some ways. So one of the things that we are thinking is encapsulating other nutrients and also maybe enzymes. Can I now deliver an enzyme in a certain spot where I want it to open up, and I want another enzyme that I want to send all the way to the theca, maybe a cellulosic enzyme or something like that. So there are ways with this technology now that you can create precision intestinal nutrition, where you can now target certain spots. If you see a problem that's prevalent, you end up with, you can target this. I wish I had a slide of it, but we just finished a trial with a broiler producer in Western Canada. They feed a lot of barley and things like that. Wet litter problems is terrible there. But with the microencapsulation, the litter was amazingly dry. The birds had feed conversion benefits of something like 8 points. We got better growth performance, better feather cover, better pigmentation. I mean, it's amazing just what gut health can do if it's precision fed in that way.
Ok, obrigada. Nós temos outra questão. O senhor deve conhecer, é o Nasser Odelata. Com as opções disponíveis que beneficiam a ecologia do trato intestinal e mantendo as interações em mente, como o senhor define qual seria a melhor combinação para ser usada?
If I knew that answer, I would be a much richer man. That is the biggest question. And my explanation is to see what the chickens tell you. Dr. Brake used to he was a colleague of mine, and he always says, the chickens will tell you what they need. And we as nutritionists sit behind a desk many times, formulating diets, trying to figure out, how to put things together. And we need to spend a lot more time in the field looking at what is the common issue and going back and saying, here's what we have. Now I'm going to create a strategic approach to deal with the primary problem and do one thing at a time. And so developing a strategy is something that this is why I spend most of my time not telling you what works, but tell how things work so you, with your creativity, can put together a proprietary solution for you. And all of the companies that supply products are part of their toolbox. So what we need to do as educators and suppliers is to educate and explain the mode of action of our products or solutions so that you can make the decision for what would be most feasible, and then to follow up with some customer service on that. I hope that's a general question, but that's the philosophy. And I found myself, if I think like an ecologist, I'm more likely to come up with a good solution than if I'm just throwing things in and seeing what happens. This would be probably a more methodical way of finding a feasible solution that's economically feasible.
Ok. Se não temos mais nenhuma pergunta local? Você tem?
Bom, aqui a gente tem um público bastante diversificado, nutricionista, pessoal de campo, né, e muitos outros. Eu queria a sua opinião, doutor, sobre, assim, hoje a gente tem um ponto, como definir os melhores indicadores de saúde intestinal, né? Porque na pesquisa já é um problema. Que indicadores a gente vai usar para avaliar essa qualidade ou a saúde intestinal que a gente quer? No campo, a gente já tem algumas ferramentas, mas eu queria a sua sugestão, né, para eles. Quais seriam, por exemplo, 3 bons indicadores de qualidade intestinal para se avaliar no campo, né? E assim, em combination, se possível.
Good question. I have worked with Elizabeth Sentin, who came up with the IC Inside model. She was one of my postdoc students years ago. And that was the purpose of going to find a biomarker. And the Animal Nutrition Consortium that I fund, more than 50% of that research is to find biomarkers in the blood or something like that. We now have companies like DSM spun out a company called Ferek based on our technology of biomarkers and combination of posting birds and trying to assess to get a better idea of gut health. But me as a farmer, okay, there's some very simple things that I look for when I go to a chicken farm. I know what a good chicken flock smells like. I know what a bad one smells like. Your nose can pick up, putrefying factors, litter condition. There's a lot of things that you can do. So if you can just learn those senses, you will be able to go into a chicken house and say, there's something going on. Remember, the other thing is the behavior of the birds. When birds start to have intestinal inset, before you see any clinical signs, they will start to vocalize more. You'll see them running around a little bit more erratically. So behavioral looking at. This is why I say you nutritionists don't sit that much behind your desk. Go look at what is normal and what is not normal. And so those are the biomarkers that I can see. The other thing is the consistency of the litter. As Dr. Fernandez showed you, If you're looking for nice white capped fecal droppings, then you're getting to a place where it looks good, right? If you see too much of the cecal droppings that don't look really dark and black, but really foamy, you've got a problem, right? And the birds are not digesting. So it's back to being like an old farmer, like my father, who would say years ago, you know, a joke was, I would tell, he said, what were you working on? This was a few years ago. I said, particle size. We're finding out that it improves gut health. And he looked at me and said, well, that's not new. And I thought, what? He says, don't you remember that every time the eggs were really dirty, I told you just throw on some whole oats or whole wheat and that'll take care of the problem. And he knew this a long time ago. Just so I want everyone, especially their new people coming into the industry all the time, you know, put aside some of your book knowledge and go back to the basics of management and smell what they see, what you feel, those kinds of things, because those are the best biomarkers I would recommend. And be skilled in those things is important. Obviously, there are other tools that help you learn those things, like the IC inside, you know, et cetera. There are some new biomarkers in blood that we can pick up. The latest one is, but it's expensive, is blood microbiome. And so you can actually say, oh, what organisms are actually coming across? And it's an indicator of leaky gut. And there's a few other symptoms to do that. I hope that answered the question.
Ok. Então, muito obrigada, professor, por ter vindo até o Brasil, né? Ele teve uma agenda muito apertada para vir aqui. Então, realmente, ele veio para esse evento, né? Tá com uma agenda muito complicada nesse final de ano, e a gente sabe que é com muito prazer que nós recebemos aqui. Tá bom? Espero que volte. Tá bem? Então,